Most of the world didn't have it until colonization started in the 15th century.
Every single day, thousands of blood donations flow through hospitals and clinics around the world. Nurses and doctors speak of blood types with clinical precision, AB, or O, as though these are just administrative categories. But behind one of these types lies a story written not in laboratories, but across the entire human genome, spanning continents and millennia. Type O blood is the universal donor. We've all heard this fact. It's the blood type taught in health class, the one that saves lives in emergency rooms when there's no time for compatibility testing. What most people don't know is that this simple medical classification conceals something far more complex. The fingerprints of ancient survival, geographic isolation, and the way diseases have invisibly shaped human genetics across vast populations. This is not a story about what makes type O blood work in transfusions. This is the story of why certain populations on Earth carry almost exclusively type O blood, while others show completely different patterns. And it all begins with a single biological question that science is only now beginning to fully answer. Let's start with the basics, because they matter. Blood type is determined by antigens, proteins and sugars that sit on the surface of your red blood cells, like tiny flags. Think of them as identification markers that your immune system uses to recognize what belongs in your body and what doesn't. Type A blood carries the A antigen. Type B carries the B antigen. Type AB carries both. But Type O carries neither A nor B antigens. Instead, it carries something called the H antigen, which is the simplest configuration possible in the ABO blood group system. This absence is precisely what makes Type O blood so universally compatible. When someone with Type A blood receives Type O blood, their immune system doesn't recognize the donor blood as foreign because Type O blood lacks the B antigen that would trigger an immune response. Conversely, Type B recipients receive Type O without rejection because it lacks the A antigen. The Type O blood simply slips into the recipient's circulatory system without setting off alarms in the immune system. This biological blank slate has made Type O invaluable in emergency medicine. In situations where a patient has lost massive amounts of blood and there's no time to determine their blood type, Type O is the default choice, the universal donor that can save lives within precious minutes. But this medical convenience masks a deeper mystery that population geneticists have been puzzling over for decades. Type O is not distributed randomly across human populations. In some parts of the world, it's relatively balanced with other blood types. In others, it reaches frequencies that seem almost improbable from a random perspective. The most striking pattern appears in the indigenous populations of the Americas. Site index equals 4-1. Type O is particularly high in frequency among the indigenous populations of Central and South America, where it approaches 100%/site in some remote Amazonian communities. The frequency reaches such extremes that Type A and Type B blood are virtually absent from the entire population. To put this in perspective, site index equals 4-1. African American populations show 47% Type O. Latin American populations show 53%. Asian populations show 39%, and Caucasian populations show 37%/site. These numbers reflect mixed populations in modern times. But in indigenous American populations that have maintained more isolated genetic structures, Type O frequencies often exceed 95%. This pattern first intrigued anthropologists and geneticists in the mid-20th century. How could an entire continent of human populations share such a uniform genetic trait? The explanation that emerged seemed straightforward: the founder effect. The founder effect is one of the most fundamental concepts in population genetics. Site index equals 4-1. This genetic phenomenon occurs when a new population is established by a very small number of individuals from a larger population. This small founding group carries only a fraction of the original population's genetic diversity/site. Applied to the Americas, the logic worked like this. If the first humans to cross from Siberia into North America happened to carry mostly Type O alleles, the genetic variants that code for blood types, then their descendants would inherit that same predominance. Over thousands of years and thousands of generations, through random genetic drift and isolation from other populations, This initial frequency could become even more pronounced. The theory was elegant and explained the data reasonably well with the knowledge available at the time. Scholars could point to the geographic isolation of the Americas, separated from Asia by thousands of miles of ocean, as sufficient to prevent genetic exchange that might dilute the original type O signal. But this explanation began to unravel when geneticists could finally examine ancient DNA directly. Around the early 2000s, advances in ancient DNA extraction technology allowed researchers to analyze genetic material from skeletal remains thousands of years old. They could now look backward through time and see what blood types people actually carried in the distant past, rather than relying on models and assumptions. The first hint of trouble came from analyzing the genetic ancestors of Native Americans. Scientists extracted DNA from ancient Siberian remains, the Vari, populations that theory said had migrated to the Americas. What they found was unexpected. Site index equals 5-1. Unlike indigenous populations in the Americas, ancient Siberians displayed a wide variety of blood types. Type A, Type B, Type O, all were present in significant numbers. This was not a small, genetically limited population dominated by Type O. It was a rich, diverse mix, much like what we see in Europe, Asia, and Africa today. Site. This discovery posed an uncomfortable question. If the ancestral Siberian populations carried multiple blood types in significant frequencies, why do the descendants in the Americas show such overwhelming Type O dominance? If genetic drift alone was responsible, researchers would expect to find pockets of Type A and Type B blood scattered throughout the Americas in various frequencies, the random remnants of lost genetic diversity. Instead, what the data showed was something that looked less like a gradual fading and more like a filter. The evidence pointed toward what geneticists call a selective pressure, some environmental or biological force that favored Type O individuals over those with Type A or B blood types. Site index equals 5-1. The overwhelming prevalence of Type O blood in the Americas could be explained if, over time, some biological force had consistently removed individuals with A and B alleles from the population, leaving only Type O survivors. This pattern raised troubling possibilities. Was there a pathogen that selectively targeted those blood types? Or was some other selective force acting on the population? Site. This wasn't speculation without foundation. Science had already documented that blood type antigens can act as attachment points for pathogens, literally the places where disease-causing organisms recognize and invade human cells. One of the most well-documented associations between blood type and disease susceptibility involves cholera. Site index equals 12-1. In people with blood type O, scientists found that cholera toxin hyperactivates a key signaling molecule in intestinal cells. Researchers found that levels of the signaling molecule were roughly twice as high in the cells with the Type O antigen than in the cells with Type A antigen, suggesting that people with Type O antigen exposed to cholera toxin would suffer more severe diarrhea. Site. This seems counterintuitive when we remember that Type O is dominant in the Americas. But the geography makes sense. Cholera isn't, and wasn't, endemic to the Americas. It's a disease primarily associated with South Asia and Africa. The more significant disease connection for explaining blood type patterns in ancient populations involves malaria. Site index equals 9-1. Blood group O is thought to confer susceptibility to diseases such as cholera and other diarrheal diseases. However, blood group O protects against severe Plasmodium falciparum malaria through the mechanism of reduced rosetting. Site. This is an example of what geneticists call a balanced polymorphism, where different alleles persist in populations because they provide advantages in different disease contexts. Type O might protect against malaria but increase vulnerability to cholera. Type A or B might show the opposite pattern. The global distribution of blood type frequencies likely represents an evolutionary balance struck over thousands of years as populations encountered different disease pressures. It's crucial to understand that genetic drift and selective pressure are not the same thing, even though both change allele frequencies in populations. Genetic drift is purely random. Imagine a small island population of 100 people where Type O blood is present in 50% of the population. By pure chance, if slightly fewer Type O individuals reproduce in one generation, the frequency might drop to 48%. In the next generation, through random variation again, it might jump to 52%. Over many generations, this random walk can cause genetic traits to be lost entirely, simply by chance. Selective pressure, by contrast, is directional. A trait persists or spreads because individuals who carry it have a survival or reproductive advantage in their particular environment. Site index equals 4-14, 4-20, 4-26, 4-32. The data indicated that the uniformity of type O across America's populations went beyond what could be expected from random drift alone. Drift can reduce diversity over time, but it rarely wipes out major alleles entirely, especially not across entire continents/site. The pattern observed in the Americas suggested something more directed than random chance. What makes the American pattern particularly interesting is its consistency. From the Arctic North to the southern cone of South America, the pattern holds. Indigenous populations, separated by thousands of miles, speaking different languages, practicing different cultures, yet they share this genetic signature. Site index equals 4-1. The most widely accepted explanation is the founder effect combined with geographic isolation. This small founding group carried a high frequency of the O allele, and subsequent events amplified this initial genetic makeup/site. But researchers now understand this explanation is incomplete. The pattern is too uniform, too consistent, too devoid of the A and B alleles to be explained by founder effects alone. What likely happened involves multiple processes working together. An initial founder population with relatively high type O frequency, then thousands of years of isolation, combined potentially with selective pressures from diseases present in the Americas that may have favored type O individuals. It's important to note that blood type is just one genetic marker among millions. Site index equals 5-1. Indigenous populations show widely divergent ABO blood group distributions. Those in Australia and North and South America almost completely lack Group B, whereas those in Asia show the highest rates of Group B. In addition, Group A is almost non-existent in indigenous populations of South and Central America, but more prevalent, greater than 30%, in Canada, Scandinavia, and Central Europe, perhaps due to selective pressure provided by smallpox/site. This last observation is particularly revealing. Type A shows higher frequencies in populations that historically encountered smallpox, suggesting that Type A individuals may have had some survival advantage against that particular disease. Conversely, in populations that never encountered smallpox, those in Central and South America before European contact, Type A didn't persist or spread. This is evolution in action, genetic frequencies changing in response to environmental pressures. Around the world, blood type distributions tell stories of migration, isolation, and disease. Site index equals 2-1. Among tribal populations of North India, type O prevalence typically ranges from 30 to 40%, which is lower than the global average. Many northern tribes show higher frequencies of type B, often 30 to 35%, which is consistent with patterns seen across parts of Central Asia. Type A shows moderate frequencies of about 20-25%. Site index equals 2-1. These patterns may reflect regional variations in blood type frequencies among indigenous populations exposed to different disease environments, with cholera susceptibility, plague resistance, and other pathogens potentially influencing population frequencies in regions where these diseases have been prevalent/site. In Southeast Asia and Africa, where malaria has been endemic for thousands of years, type O shows lower frequencies than in the Americas, despite some genetic connections through migration. This reflects different selective pressures. Populations in malaria zones experience selection that may have favored individuals with other blood types. Understanding blood type distribution isn't merely academic. It reveals how humans adapt to their environments at the genetic level, how diseases invisible to our ancestors shape the genetic diversity we see today, and how geographic isolation creates and maintains genetic differences between human populations. The story of type O blood in the Americas is not a story of mysterious disappearances or hidden mechanisms. It's the story of how a small founding population, isolated for thousands of years, maintained and amplified a genetic trait through both chance and potentially through natural selection. It's a story written in geography, disease, and time. Modern genetics has given us the ability to read these stories written in DNA. Site index equals 5-1. The vanishing of A and B in the Americas was not the sort of genetic change that could be readily explained away by reference to geography, climate, or chance alone. It suggests a powerful filter through which only one blood type passed intact, /site. But that filter wasn't mysterious or engineered. It was the natural product of human history, migration, isolation, and adaptation to local disease environments. Type O blood is indeed the universal donor in the modern world, where transfusion medicine has transcended the need for genetic matching. But in the ancient world, it may have been something else, a marker of adaptation, a genetic legacy of survival, a trace of how our ancestors lived and died in environments we can barely imagine. The science tells us that human genetic diversity, far from being random, reflects the deep history of our species, where we've been, what diseases we've encountered, and how we've adapted. Type O blood in the Americas is not mysterious. It's a textbook example of population genetics at work, written not in laboratory notes, but in the genomes of millions of people. Understanding this, understanding that genetic patterns tell stories of real human history, is perhaps the most important scientific lesson we can learn from blood type distribution. It teaches us that human biology and human history are inextricably intertwined, and that the science investigating this connection is one of humanity's most valuable tools for understanding who we are and where we come from. Our Environmental Literacy Council. Why are Native Americans Type O blood? 2025. PNAS. Proceedings of the National Academy of Sciences. Blood group O protects against severe Plasmodium falciparum malaria through the mechanism of reduced rosetting. 2007. Our Washington University School of Medicine. Study may explain why people with Type O blood more likely to die of cholera. 2016. ResearchGate. Blood group O alleles in Native Americans. Implications in the peopling of the Americas. 2009. BA Notes. Geographic distribution of ABO blood groups among indigenous peoples. 2025. NCBI/PubMed. Multiple peer-reviewed sources on blood group genetics and disease association. Open Boston University. Blood group O and risk of infection with Vibrio cholerae. Academic research compilation.